Okay, so D4 is the option unit, which is very, very closely aligned to 6.2. So the first thing I'd ask you to do is pause here, go back to the 6.2 notes and check through that particular booklet and what's there. These are the syllabus statements for understandings. This is what most of the questions are going to be targeting, but then we also need to consider these applications and skills after that. So next test, if you like, can you fully draw and label the structure of a heart? Can you show the direction that blood flows through the heart? These terms at the bottom here, and many of these are ones that you ought to be familiar with. So again, I'd pause the video here, I'd have a go at drawing that before moving on. Hopefully this is something that you've managed to construct. And just to make sure that we appreciate that the heart is separated down the middle, this is the septum here. On the top, we've got the atrium in this particular region here, and the pulmonary vein. We've got two of them, one from each of the lungs, is going to pour blood into here. When there is sufficient blood inside, it's going to push open these bicuspid valves. The tissue around here, the cardiac tissue, will contract. That puts the blood under high pressure here, and it's going to push it through those valves and into the bottom chamber. The bottom chamber is the ventricle. The ventricle also has got this muscular layer here, is cardiac muscle. When that contracts, it's going to push blood under high pressure upwards. The contraction happens from the bottom, from the apex here upwards. And when it goes upwards, it's going to push these valves closed, which means that the blood can't go backwards, and instead it's going to redirect blood up through here, pushing open the semilunar valves and out through the aorta. The aorta is the biggest artery in your body, and that's going to branch to feed the various organs of your body. So again, let's look at this once more. You've got the pulmonary blood coming in. This is oxygenated blood. It fills up this chamber. It pushes open the bicuspid valves. The contraction of the atria, also called atrial systole, is going to force blood down into the ventricle. Ventricular systole, or the ventricle contracting, is going to put blood under high pressure, push open the semilunar valves and push blood into the aorta. Now, blood is always going to flow from an area of high pressure to low pressure. If we look at the opposite side, the right side, same sort of idea. We've got blood draining from the body in these great veins, or the vena cava. The blood flows into the right atrium. When you've got significant quantities of blood in there, that will push open these tricuspid valves. That will allow the blood to flow down pressure gradient into this bottom chamber, the ventricle. You've got, again, atrial systole, ventricular systole. The ventricle is going to contract from the apex upwards, pushing blood upwards in this direction, closing the tricuspid valve, pushing open the tricuspid valves and pushing it back up to the pulmonary artery. This is going to be deoxygenated blood flowing in this direction. Now, hopefully that's all pretty clear. Just a word of note, the atrioventricular valves are exactly the same thing as the bicuspid and the tricuspid valve. This is one sort of overarching name for both of those. So the bicuspid valve is found on the left side and the tricuspid on the right side. If we look at this diagram, this is again showing you much the same thing. It's a little bit more detail, and if you cast your minds back to the heart dissection that we did, we noticed these blood vessels on the surface here. These are your coronary arteries and veins, and the coronary artery is going to bring oxygenated blood to the muscle, the cardiac muscle. Notice also here this left side is significantly thicker compared to that of the right, and also significant is that both ventricles are much thicker than the atrial walls here. Using the numbers, okay, that are on here, I would again suggest that you talk this through and make sure that you are clear about the naming of the different parts and the blood flowing in the direction moving from a higher pressure to a lower pressure. Here again, you've got certain things which are hopefully familiar. The red is denoting oxygenated blood. That is blood with oxygen bound to the hemoglobin within the red blood cells, whereas the blue instead is deoxygenated. You've got the atrioventricular valves, bicuspid and tricuspid. We've mentioned the semilunar valves, and the semilunar valves are also called the pulmonary valve or the aortic valve, depending on where they are. So the pulmonary valve here and the aortic valve at the base of the aorta. Now, cardiac muscle is described as being myogenic. Now, this is interesting because it means that the heart can beat without there being a direct nervous system to the heart to start that contraction. So if you take a heart out of a body and you place it into something called Ringer's solution, which is similar to tissue fluid, it will continue to it will continue to beat for a period of time because of this idea of myogenicity. And what we need to do in Unit D is understand how that actually operates. To do so, we need to think a little bit about the structure of the heart. This diagram is reminding you that there are three different types of muscle. We've got skeletal muscle. They're the muscles that move the skeleton of your body, your bones. We've got smooth muscle. Smooth muscle is found inside the arterial walls. It's also found in your digestive tract and various other places in the body. And then the cardiac tissue, which is the tissue of the heart. So cardiac muscle tissue is the tissue of the heart. Now, you don't need to know too much about the differences between these, but you do need to be aware of the special features of cardiac muscle and how the structure relates to the function of this one in particular. So what we've got here are a series of cells which have got these sort of branching structures here. Now, these branches is not possible to see this on the actual diagram, but these are moving in 3 dimensions. And the purpose of these cardiac muscle cells being branched is that allows faster signal propagation. So we're going to talk about the fact that a wave of electricity is going to pass across these cells, and when that happens in the atria, they all contract, and this allows that propagation of the signal all the way through the muscle tissue. What we've also got at this particular area here, you can see an intercalated disc. Now, in many cells of the body, you've got tight junctions. That is that the two pieces of the cell are very closely associated with one another. Now, in cardiac muscle cells, you've got gap junctions, and gap junctions are here at this intercalated disc area here. So they're not fused in the same way as they are elsewhere. What you also notice is that there are many mitochondria, more mitochondria in cardiac muscle cells compared to smooth muscle or compared to skeletal muscle. And that's very important because the heart has got this property of myogenicity, but the other property that it does have is that it doesn't fatigue. So it beats for 24/7 and it doesn't fatigue, which is pretty unique. And that is also to do with the structure of this tissue. We look at this next one, we need to appreciate what the actual cardiac tissue is, what its structure is, what its function is, why it's so special. So again, it's special because it's got this ability to be myogenic and it doesn't fatigue. Cardiac muscle might also be called cardiac and heart muscle is interchangeable words, or myocardium is also referring to exactly the same thing. And what we can notice here on this diagram is this branching structure, which, as I said, allows the propagation of signals to move across. Now, for higher level, they need to make sure that they read this in a lot of detail because they do need to look at the skeletal muscle in Unit 9. For standard level, we don't need to pay so much attention to this. Things that you might want to be focused on here is this idea that both of them are made of fibers, and the fibers are called actin and myosin fibers. They are multinucleated. Remember, we met this idea in Unit 1 because it's unusual because you've got it doesn't conform to the cell theory. The striations is the stripy appearance, and that's true of skeletal muscle and cardiac muscle. T-tubules are invaginations, membranous invaginations, which allow, in particular, calcium to move, which causes the muscle to contract. Again, this part and the sarcoplasmic reticulum is a developed endoplasmic reticulum. It's only important for those of you that are in higher level, not at standard level. So we've got, I think, read that. Anything that pops up that you're not sure of, then let me know. The motor function here is movement, is voluntary movement for skeletal muscle, whereas in cardiac muscle, we don't have to think about it. It's not voluntary. We can't make it stop. We can't make it go faster by will. It's an involuntary muscle, and that comes from the change of the pace of the heart comes from an autonomic input. So the heart is myogenic. It beats by itself, but the speed at which it beats is going to be dependent on the autonomic nervous system, which we'll come back to later on. This slide is again showing you the same sort of idea, the branching structure of the cardiac muscle. It's multinucleated. It's got these invaginations. So we've got the T tubules here, and the gap junctions are those loose junctions. Many, many, many mitochondria. So have a read through this. And again, anything on there that you don't understand, you need to come back to me and ask. This is here principally for the higher level students, but again, it's worth you having a look at. They could give you an electron micrograph and they could ask you to identify certain features like an intercalated disc. That has come up as a past paper question. So remember that the heart is myogenic. The branching structure is three-dimensional branching structures, and that means that you can get fast propagation throughout the entire muscle tissue. You've got these intercalated discs. They're not fused tightly together. You've got many, many mitochondria. Incidentally, the intercalated discs are the same thing. That allows an easy flow of electricity between them. Many mitochondria, which prevents the muscle from becoming fatigued. And the mitochondria also signal into you the fact that there must be aerobic respiration, so respiration with oxygen within cardiac tissue. One thing that cardiac tissue doesn't do is become fatigued. And the reason it doesn't become fatigued is there's something called a refractive period, which is long. Now, again, for standard level, you don't have to know about the refractory period. So I wouldn't worry too much about it. But this is why the muscle doesn't become fatigued. That is what you certainly do need to know. Here again, for higher level, it's important, but they could ask you to name certain things. They won't ask you about the sarcomere or the Z line, okay, but they might ask you to identify a gap junction.